Okay, so chapter 3, it's about microscopy. So in this chapter, we're going to learn what are the units of measurement to measure microorganisms. We are also going to see some of the equivalents, and we're going to see the different types of microscopes that we have and how do they work and what are their uses. As well, we're going to see what is magnification and what is resolution, which are terms that frequently we use when we speak about microscopes. So this table is showing you some of the units of length and their equivalent in the in the metric system and the equivalent in the US equivalent system. So here we have in the left column the metric units. We have here some prefix, meaning like that's the first part of the word that we use to tell what is this unit. And then what is the metric equivalent and what is the US equivalent. So for instance, a kilometer, which is written with km, it has the prefix kilo, which means 1,000. So a kilometer has 1,000 meters, as you can see here, or 10 to the 3, to the power of 3 meters. The equivalent of 1 kilometer will be 0.62 miles. So in 1 mile, we have 1.61 kilometers. And for a meter, the abbreviation for meter is M, lowercase m. And then this is the standard unit of length in the metric system. I mean, meter, basically the system metric means meter. And then in 1 meter, we have 39.37 inches or 3.28 feet or 1.09 yards. So 1 decimeter or dm with lowercase again, it is 1/10 of a meter. Deci is 1/10. So it will be written 0.1 meters or 10 to the minus 1. Anytime that you have a point and then you have a number to the right of that point and you have this number that means something, well, in this case, 0.01, which is 2 numbers after the point, means that you have 2 units, or 10 to the minus 2. So 1 centimeter is equivalent to 0.39 inches, or 1 inch is equivalent to 2.54 centimeters. Now, in science, we use the metric system because it uses units of 10. So it's much easier to make calculations and make less mistake with units of 10 than without them. So then, in every case for the column that we see here, our units are getting a little bit smaller. In this case, like 1,000 times smaller. And then in the case of the meter, we have 10 decimeters. So it's basically 10 times smaller. And now 1 centimeter is 10 times smaller than the decimeter because we have 10 centimeters in 1 decimeter. And then when it comes to millimeters, we have 10 millimeters in 1 centimeter. And then the unit it is written with mm, lowercase, both of them, and milli means 1,000. So we have within 1 meter, 1,000 millimeters. And you can write it down as 0.001 meters, and it can be abbreviated as, or written in scientific notation, 10 to the minus 3. And there is no equivalent of US equivalent for the millimeter, because it's very tiny. Now, for micrometers, micrometers are abbreviated with the Greek letter mu and then an M. And the prefix for micrometers is micro, which means 1 millionth. Is 1 over 1 million. So it's 1 millionth of a meter. So we have 1 million micrometers in 1 meter. And then the way that you write it down in scientific notation is 10 to the minus 6 meters. And how do you write it down? Well, with a 0, a dot, and then 5 zeros after to the right of the dot, and then 1. So basically 1 micrometer will be 1 times 10 to the minus 6 meters. And again, we don't have US equivalent because the unit is very small for measuring in inches, which is the metric unit of US. And then 1 nanometer, or nm with lowercase, both of them, it is basically 1 billionth of a meter, or basically 1 divided by 1 billion, which has 9 zeros. And then it will be, write it down like 0 dot, and then you have 8 zeros after that, and then the last one will be 1. Or will be equal to 10 to the minus 9 meters. For picometers, so it will be PM, and it is very small, the unit. So basically it is 1 divided by, I cannot say this number, but basically it can be a trillion, yes, because it has 12 zeros. So, and then when you write it down in long notation, basically you have a 0, a dot, and then 11 zeros and 1 at the end. Or can be abbreviated in scientific notation, 10 to the minus 12. So there are some items in our world, like objects that are very small, and they can be measured with picometers or nanometers, micrometers, millimeters. But this is like kind of one of the smallest units that there are. And then I wrote down in here the most common types that we use for microscopy: micrometers and nanometers, and their equivalent in meters: 10 to the minus 6, 10 to the minus 9. So we have, again, in 1 meter, we have 10 to the minus 3, or 1,000 millimeters. So 1 millimeter equals 10 to the minus 3. And 1 millimeter equals 1,000 micrometers. So this unit is bigger than this, because this one will be 0.01 micrometers. And then we will see over and over again in some of these images from microorganisms how in the images they provide you a scale and they give you this abbreviation in that scale, meaning how big can be that object when you move that bar, scale bar, into the image so that you can observe it easily. So here we have these different objects, and like we have a thick red blood cells, E. coli, which is a bacteria. We have a virus, which is, in this case, T-even viruses, and then we have the DNA. So in this left side, they show us like the different scales in metric units, and then it tells you what you can see with these scales. Like for instance, with an AI, we can see objects greater than, this is the symbol, greater than or equal to 200 micrometers and above. So greater than 200 micrometers or equal to 200 micrometers, you can see objects without any help of a microscope, a naked eye. And of course, I mean, when you're young and you don't have visual problems, it's much easier for you to see really small objects. But of course, as we age, we have to wear lenses, glasses, and in this case, you can see. So then this is a thick that can be seen with an innate eye, and it's less than 1 centimeter. So this is like the scale between 200 micrometers up to 1 meter or more, of course. But basically what it tells you here with this line is that this thick can be maybe like 2 or 3 millimeters, and you can see it. When it comes to red blood cells, you cannot see them without the aid of a microscope, the complete cells. So in here, in this little square, it tells you what type of microscope we are using, which is, in this case, is LM, for light microscope. And then in here, they have this bar scale that tells you that the distance between the beginning and the end of this bar scale, this will be 5 micrometers. So if you extrapolate or bring this scale, like put it next to this object, it can tell you more or less that one erythrocyte or one red blood cell, it has a side-to-side diameter of 5 micrometers. This, it's a meter. So it's like if it was this bar. If we put it like here next to me, you can tell that I'm taller than 1 meter. Or you can go up to here, and then you can say, well, she measures 1 meter and actually 54 centimeters, 1.54 meters. You want to measure me side to side, you use the same scale, and then you can tell, of course you start with the 0 here, and then my side to side will be like kind of 42 centimeters. So that's why you use these bar scales in here. And then for these red blood cells, you can see it with these light microscopes that can see objects between, help you to see objects, between 200 nanometers and 10 millimeters. So this is what these light microscopes can see. So they can see red blood cells, they can see E. coli cells, but they cannot see these viruses. The viruses are very small. Now, E. coli, which with this bar scale, which is in this case micrometer still the unit, if you measure it in length, more or less, like in this case, this tiny one, this one it's about to divide, this one is like 2 micrometers in length. And then in width, if you extrapolate, well, it can be like 1 micrometer. And then E. coli, you can see it with the light microscope or also with some specialized microscopes that are called a scanning electron microscope or SEM or transmission electron microscope or TEM. And a scanning electron microscope allows you to see objects between 10 nanometers to 1 millimeter. Transmission electron microscope between 10 picometers and 100 micrometers. And then these bacteriophages, which is a virus that eats bacteria. So basically, this virus, it is infecting these cells. And this is how they look. They look like little alien chips. So this scale in here is 50 nanometers, which is very small in comparison to a micrometer. So if you extrapolate this and you measure the pins that are landing into the surface of this bacteria, you can say, well, the width from one pin to the other one is 50 micrometers. And also in this case, for this one, the head, which is this part of the virus, it measures from side to side 50 nanometers. And then in here, it tells you what microscope you're using, TEM. This one was SEM. It lets you to see three-dimensional view. This one lets you see internal structures, TEM, in a very thin film. And then these viruses, which are DNA viruses, you can observe the DNA with atomic force microscope or AFM. And then in this case, the bar scale is 10. So if you extrapolate this bar into here, or you can say, well, this piece of DNA measures 10 micrometers, and this is double helix DNA. So an atomic force microscope lets you to see objects between 0.1 nanometers, which is 1/10 of a nanometer, and 10 nanometers, which is very small. Any questions? So anytime that you see a color box in red color, that means that it's a microscope, that it is not light microscope. And when you see that dark box with LM, that means that it's a light microscope. So let me change this a little bit. An exercise of how to measure. So again, these are the 2 units that we use the most to measure these small objects in microscopy, micrometers or nanometers, which is 10 to the minus 9 meters or 10 to the minus 6. So if you are measuring a microorganism and the microorganism has basically, let's say, 1 micrometer, you can say that it has 10 nanometers, or if it's 1 nanometer, you can say that this microorganism has 0.001 micrometers. So 1,000 nanometers equals 1 micrometer. And something that you can see here, it's a scanning electron micrograph of this curved-shaped bacteria with an extension that is called flagella. So maybe someone will be measuring the diameter of the flagella or at the tip of the flagella, and maybe it would be 1 nanometer, which is 0.001 micrometers, or 10 to the minus 3, which is, the unit. Now, in optical microscopy, we have simple microscopes that uses one lens, or we have compound microscopes that uses 2 lenses. And with the magnification of these 2 lenses, you obtain a higher magnification. So, this microscope replica of Antoine van Leeuwenhoek, how would you classify it? As simple microscope or as a compound microscope, based on the number of lenses? People who wear glasses. You have a microscope, basically. Is it simple or is it compound? It's simple because you only have 1 layer of lenses. Now, if you use a magnifying glass and then your lenses to magnify the object, well, you will see it better, and that kind of be like an artificial compound microscope. Okay? So the microscopes that we use in the lab are compound microscopes because they have more than 1 lens. So these are the examples of different types of microscopes that we have. We have compound light microscope, we have dark field, we have phase contrast, differential interference, fluorescence, and confocal. So we will see the uses of each of these microscopes. Here are the parts of a compound microscope. The compound microscope has ocular lenses, which helps to magnify more the object as is magnified by these objective lenses. The objective lens magnifies a specific number of times, between 5 to 100 times the object, and this one typically magnifies the object 10 times. So it's just a matter to calculate the magnification, or how big you will see the object, of multiplying the number of times that ocular lenses magnifies the object times the objective lens. So the minimum magnification that you can obtain is 50, because you multiply the times that this small objective lens magnifies, which is 5 times 10 times this. So 5 times 10, 50. The next objective lens, which is the second largest, magnifies 10 times. So 10 times 10, the total magnification will be 100. The third longest magnifies the object 40 times. So 40 times 10, that's 400. And then the last one, which is the longest, and you always use immersion oil when you have it, in this case, it magnifies 100 times. So 100 times 10, that is 1,000. So that's the maximum number of times that you can see the object magnify. So that's the ocular lens, then magnifies more the image as is coming from this objective lens. And then this is the eyepiece that holds this ocular lens, and here we have the body tube that transmits the rays of light from the objective lens into the ocular lens, and then this is the arm that you use to hold the microscope as you are transporting it or moving it within the countertop. And then this is the nose piece that holds the objective lenses. The objective lenses have different length and they have different resolution, which is the ability to distinguish the distance between 2 points, where you see 2 points clearer. The higher the resolution, the sharper the image that is going to be, because you can distinguish better where 2 points are located close by. So they have a line, color line around it that tells you what is, if you know that code, what magnification. So 5 is like the one with the red line around, 10 with the yellow line around, 40 with the blue line around, and 100 with the white line around. And they're hauled into this nose piece. And now this is a stage that has clips that holds the specimen, the slide with the specimen. And then underneath the stage, we have condenser lenses that focuses the rate of light into the middle of the objective lens so that the rays of light are not lost. The rays of light are not lost. And then we have underneath this condenser lens a diaphragm, which is basically like an object that can open and close, just like the iris in our eyes, that allows light to go through more or less. So when you close the diaphragm, less light goes through the pathway of light. But when you open it, more light goes through. So that's the diaphragm. And then we have these mechanical knobs that can move the stage front and back and side to side, so that whenever you're looking to a microscope object, you can scan it. And then at the base of the microscope, which is this, you have a light switch, and then you have this sliding knob that basically controls the amount of light that goes through the lamp or illuminator, in which you have a light bulb, or you may have LED that allows light to go through the object, and then you will be able to see. And then on the sides, you have here 2 knobs. One that is small, that is called the fine tuning or fine focusing knob, and then you have the coarse focusing knob, which is the largest. This one lets you to move the object, the stage in smaller distance up and down, while this one lets you to use it in bigger distance up and down. So tomorrow we will have the light microscopy lab, and then you will practice focusing objects with the microscope. Most of you already have used it before, but no matter how experienced someone is, we always benefit from practicing more. So, a total magnification in microscopy, it is obtained, in the case, of course, of light microscopy, by multiplying the magnifications given by the objective lens times the ocular lens. And again, the term resolution means how easily a lens can distinguish between 2 different points. It's like the pixels in cameras. The higher the pixel, the better the resolution it is, the sharpness that the object will be. So a microscope with a resolving power, that's the other name for resolution, resolving power, of 0.04 nanometers, can distinguish between 2 points that are farther apart, greater than or equal to 0.4 nanometers. So this is a good resolution. And then when you have an object hit by these light waves, the shorter the wavelength, the greater the resolution. So for instance, you can have two microscopes, microscope number 1 and microscope number 2. Let's say that microscope number 1 doesn't have as much resolution as microscope number 2. So you have light wavelengths that will go like this and hit the object. So in this case, for microscope number 1, your object is going to magnify, but it's going to look blurry. With microscope number 2, in which you have better resolution, the wavelengths are smaller. So the distance between one part of the wave and the other one, this one is bigger than this. So in this case, you will see sharper the object with a better resolution microscope. So in this, well, you lose the shape, while this one you can see it very clear. And this is the pathway of light of the microscopes. So pretty much compound microscope, everything starts at the illuminator, passes to the diaphragm, the condenser lenses, and then into the specimen. So what the condenser lens do, because there are several rays of light, what it does is basically focus it into a particular area in the specimen. And then as the rays of light goes into the objective lenses, as they hit these objective lenses, now they will be separated again. And then inside this body tube, you have a prism where you, like if it was like a mirror, it hits these rays of light, this prism, and then this rays of light, are redirected into the body of the lenses, or like the eyepieces, and then into the ocular lenses. And then you can see it through these ocular lenses. So that's the path of light. And now we have photons of light in our environment that hit objects, and then it provides this refraction or bending, which help us to see the different colors and the different shapes of the objects. So we have in microscopy another term which is called refractive index, which is basically measuring the ability of light to bend in a medium. So light may bend in air so much that it misses the small high magnification lens. So in this case, when we're looking at objects in the microscope, as we move from the smallest objective lens into the largest, which is 100 times, or the immersion oil objective lens, you can lose rays of light, but you will use this immersion oil that will be on top of this glass slide, that it has the same density as the glass, so that you don't lose as much rays of light. So this is how it looks like. If you will be looking at a magnification in this area, where you have the stage and the specimen, so this is what it will happen. So you have here two rays of light. And of course, this is just for illustration purposes. So here we have this red line that represents a type of ray of light that it hits the condenser lenses that typically focus it here, or condense the rays of light into here. This is the glass slide on top of this stage. So the stage is here. And then, if you have an objective lens that is, of course, immersion oil, and you didn't add the immersion oil, these rays of light will be lost. As these rays of light hits this glass slide, they will be bent through refraction, refraction, is called bent. So they will bend, and then they will be lost because they don't follow the pathway where you have the objective lens. However, if you put immersion oil, for instance, this blue ray of light, it will be focused here into the specimen, and then it will follow its pathway, refracting a little bit, but this immersion oil, since it has the same refractive index as this, it will prevent so much bending, and it will be catched by this objective lens. So now we don't use immersion oil in every single objective lens, only in the ones where it's needed, which is like the 100 times. So then for microscope types, so we have bright field, we have phase contrast, and we have dark field. With bright field, you have the object surrounded by a bright field, like clear field around the object. The object is going to look dark. And then the light reflected off this specimen does not enter the objective lens. Typically, the objects are not going to be live, are going to be dead, because you, in the case of bacteria, you treat them properly so you don't have live bacteria or protozoans. But sometimes you can use light microscopy to see light objects, but it's not going to be very good as when you use phase contrast. Phase contrast is going to basically have the dark background, and then the object is going to look clear or transparent. And then the light reflected of the specimen enters the objective lens. Now in dark field, it's going to be the opposite of bright field. So you will be seeing a dark background, and the object, most of the times it is stained with a type of dye that is called fluorescence. So you will see like fluorescent light, and then will be surrounded by a dark area. So here are the different types of microscope then. This specimen here is paramecium. It's a type of protozoan. So here we have paramecium seen more or less, it's unstained, so it looks more or less transparent. You can see the internal part, so this is the cytoplasm. Protozoans are unicellular organisms, and all of these are pili that surrounds this plasma membrane. And then this is basically dark field, and here is phase contrast. So in phase contrast, what you do is you make the diffraction to be more accentuated as the light passes through the specimen, so that you can see the internal components. Like these protozoans, they eat food that they encounter in the environment, and you can study the transit of food through the cytoplasm using this phase contrast. DIC is similar to phase contrast, but uses two beams of light. So that gives you better resolution. So it gives you like a three-dimensional shape. So here is an image of this protozoan as seen with this DIC, which stands for differential interference contrast, by using 2 photons or 2 rays of light. So all of this is the cytoplasm, and these round objects, these are vacuoles. And these are pili that surrounds this plasma membrane of this protozoan. Fluorescence microscopy enhances the view of objects by staining the object with fluorochromes, which are dyes that absorb rate of light and emit a higher energy. And then you have to use UV light to excite these fluorochromes so that you can have fluorescence. And then you can have fluorescence with different colors. Sometimes you can use green, blue, red, orange. And in this case, for instance, you are staining a type of bacteria that is called treponema. This bacteria has a spiral shape, it's very thin, and if you try to stain it with a conventional technique of staining microbes in microscopy, which is gram staining, you can damage these cells. So that's why you have to use fluorescence. And fluorescence is actually very specific and sensitive, so it's very nice. And then when you use antibodies to detect the presence of these surfaces, proteins in the objects, you have something that's called immunofluorescence. So it's very useful to detect proteins in tissues or clinical specimens. Now we have a different type of microscope that is called confocal that also uses UV light. And in this case, you are sending these rays of UV light into different levels in the specimen, in layers. And then you use the software in a computer to basically put on top of each other these stacks of images, and then you can recreate this object and look at it as if it was a three-dimensional view. So this is the previous specimen, but now seen with confocal. So we have used, in this case, 2 types of fluorescence or fluorochromes, like in this case, orange for the pili and also for the nucleus, which is seen in here, and green for the cytoskeleton. So you see more vibrant objects with this type of microscopes, and it uses a single photon of light, and it can basically take images of one planet at a time up to 100 micrometers in depth. Like if you use your phones to take pictures. Well, you can start taking pictures of my feet and my legs, and then you move up to my head, and then you put them all together, you can recreate a three-dimensional image with that software. Now, we have 2-photon microscopy, in which you use 2 rays of light, or 2 photons, and it can image living cells up to 1 millimeter in depth. So these photons are not as strong, and it helps you to see the activity of the cells in real time. Like in this case, this paramecium, that it can be seen processing food. So these are food vacuoles, like little pouches with food. And then this is the cytoskeletal proteins. This is the pili. And what is this? That's the nucleus. Now, we have a type of microscope that lets you to see the surface of objects with the tip that they use. So they basically scan the surface and let you see this three-dimensional view of very, very tiny objects. So this is called a scanning acoustic microscopy, or SAM. So it uses sound waves, and these sound waves hit the object, they get reflected back, the tip catches this energy, and then with the software, you can produce these three-dimensional images. Like it can be the surface of cancer cells, the plaque in an artery, which is basically an obstruction inside an artery, or it can be a biofilm. The resolution is very high, up to 1 micrometer. So these are very nice microscopes. And now, for electron microscopy, which are very sophisticated microscopes, you use electrons to hit the object. You use a beam of electrons, hit the objects. They're catched by detectors of these rebouncing electrons, and then you can obtain high resolution of internal structures or of the surface of these objects. So you can have a resolution of 0.2 micrometers with these electron microscopes. And we have transmission electron microscope, or TEM, and you have a scanning electron microscope, or SEM. A scanning electron microscope lets you look at the surface of an entire object, while the transmission electron microscope lets you to see the internal structures of a thin slice of a specimen that you have there. So TEM uses ultra-thin sections of the specimen, which sometimes can be coated with salts of metals, heavy metals, to prevent damage of these structures, because heavy metals resist more the heating of electrons. And then with these, you can magnify up to 100,000 times at a resolution of 2.5 nanometers. So what are the uses of TEM? You can look at viruses, which are very tiny. So you can magnify a virus up to 100,000 times. Or you may want to see organelles, like what if you want to see the mitochondria that roughen the plasmic reticulum, the nucleus. So you can use these microscopes. And again, you use a very thin section. So look at this paramecium with these microscopes, which are electron microscopes. So they have an electron gun, they have an electron collector, they have an amplifier, and then they send this image into our software so that you can have this type of view. This was very likely coated with gold, and if not, gold salts. And I know it's very expensive nowadays, the ounce. It's 10 times expensive than what it used to be like 40 years. Anyway, so you can see better resolution of the internal structures, and here's the nucleus. Scanning electron microscope, it gives you like the three-dimensional view of the surface of a whole specimen. And it uses electrons just like the other one. And then in this case, the scanning electron microscope goes side to side with this gun of electrons hitting the surface. And then it magnifies less than the TEM, Like, look, it can magnify it up to 10,000 times. But it is good. I mean, it has a resolution of 20 nanometers. And look at how it lets you to look at these images. It's the same as before that we have seen over and over with the different microscopes. But this paramecium, now you can see the surface. You can see like the little peeling. So it looks like a shaggy surface. The same deal, has an electron gun, has collectors of electrons, has amplifiers, send this image to a software, and then you can enhance the color with this software. Maybe the other one was enhanced with color, not necessarily need to be coated with gold. And gold is an example. They can use different salts. Now we have scan probe microscopy. Like in the case of the scanning tunneling microscope, or STM. So you have a metal probe, it can be tungsten, for instance, and that goes over the surface of the object and reveals the topography if you have bumps, depressions in this surface. And the resolution is 1/100 of an atom. So it lets you see atoms, basically. Give you the view of molecules inside, like the DNA or proteins. So this can be a protein inside this paramecium. Lipids. Atomic force microscope, another scan probe microscope. So it uses metal or diamond probes that are inserted into the specimen, and it gives you a very high resolution. So this will be porins in a surface, in the plasma membrane. These microscopes are very expensive. Not everyone can use these microscopes, only someone who is highly trained. But it gives you very good resolution. I mean, what if one of these channels or these porins was mutated and is closed? You can see it with this type of microscopes. So that's all about microscopy. Do you have any questions? Okay, now let's look at the preparation of these specimens for light microscopy. So we're going to see the different types of dyes that we have, including acidic and basic. We're going to see specific staining techniques like gram stain, simple stain, and acid-fast stain, and structural stains as well, like capsule, endospore, and flagellar stain. So staining basically is the technique that it is used to have visible objects available to look with the aid of microscope. Most of the times these objects, they don't have fluorescence of their own, or they cannot be seen because they are transparent without the aid of dyes. So in this case, you would use chromophores or dyes. And the chromophores, they have different names, and they have different electrical charges. And with these electrical charges, you can have interaction of these dyes with the object, and they can either go inside into the object and stain it internally, or they can stay outside. And then you can see the surrounding outside this object. Now, you have to prepare this object by producing a smear. What is a smear? It's a very thin film. And this smear will be added into a glass slide. And typically this smear, it is passed through a flame twice or 4 times really quick to fix the object or your specimen into the glass slide, and in the case of bacterial samples, to kill the microorganism and adhere it into the sample. Because as you are treating this with the different chemicals, with the different dyes, and you rinse it, there is the risk of losing this specimen if you don't fix it. Fixation is basically passing this glass slide with the specimen twice or 4 times really quickly through an open flame. And this open flame doesn't have to be so high that you will destroy the cells that makes your specimen. And that's why you also pass it quick. No matter if it's a very light flame, if you don't pass it quickly, or if you leave it there for a long time, of course, that heat is going to destroy your specimen. And you will learn how to produce films when you will be doing simple stains and then differential stains. So then live or unstained cells, they have little contrast with the surrounding medium, so that's why you have to use these dyes. So you have positively and negatively ions or charged ions in these dyes. So these dyes are made of positive or negative charges with these ions that makes the dye. So a basic dye has a positive charge. Positive charge is a cation. A molecule that has a positive charge is a cation. The cells are negatively charged. So a basic dye will be attracted into inside of the cell, the cytoplasm, by the negative charges that these have. So a basic dye will stain the cytoplasm of the cells. Now a negative charge chromophore or dye has negative charges, has ions that are negatively charged, which are called anions. And these anions won't penetrate the cells because they are negatively charged, because of the law of the opposite attracts. So negative is repelled to negative, positive is repelled to positive, and positive is attracted to negative, negative is attracted to positive. So if you have a negatively charged molecule or dye, and your cell in the cytoplasm is negatively charged, it won't penetrate. So an acidic or negative charged dye will stain the surrounding of the cell. So here is an illustration about this principle of staining. So here we have in this left side an acidic dye, pH 5, and here on this side we have a basic dye, pH 8. A basic dye has positive charges, so this is a cation. An acidic dye has a negative charge. Since the cells are typically negatively charged, if you put an acidic dye, this acidic dye will stay surrounding the cell. In this case, this will represent the plasma membrane, the dashed lines, and this will represent the cytoplasm. So the cytoplasm or the cell will be colorless, and you stain the background with the color of your dye. In this case, for a basic dye, this dye will start converting these negative charges into positive as it's coming and interact with them. Or at least you will have more positive charges when this dye penetrates, and then the cell will be staining the cytoplasm while the surrounding is going to be colorless. So that's for a basic dye versus an acidic dye. Now, depending on the number of dyes that you use to stain an object, you will have a simple stain or a compound or differential stain. If you use one single stain, that will be simple stain. If you use more than one, it will be a differential stain. So In simple stains, sometimes we use, and even in differential stains, we use mordants, which are chemicals that helps to retain the dye inside the cells. So it helps to hold the dye inside the cells, or can be used to coat the specimen to enlarge it. That's the use of mordants. So now, in differential stains, we use 2 or more dyes. When you have more than one group of cells, to differentiate the different types of cells. So examples of differential stains will be gram stain and acid fast, which help us to distinguish different groups of bacteria. And then for special stains, it helps to stain something that is not the cytoplasm. It can be staining the capsule, which is found outside the plasma membrane of the cells, or it can help you to stain internal structures like endospores, which are dormant bacteria that are formed by cells before the cells die. Or you can stain extensions that the bacteria uses in the cell wall, extensions of the cell wall that the bacteria uses to move, like flagella. So in this case, you use spatial stains or structural stains. So for gram stain, so basically this was the first technique that it was used to distinguish groups of bacteria. And this bacteria that is stained with this technique can be classified as gram-positive bacteria or gram-negative bacteria. Gram-positive bacteria stains purple, while gram-negative bacteria stains red. And gram-positive bacteria tend to be killed by antibiotics like penicillin and also by chemicals that we know as detergents. While gram-negative bacteria is more resistant to antibiotics and detergents. Gram-positive stains purple because the primary dye, the first dye that you use, it is purple in color. It's called crystal violet. It looks violet. Now, gram-negative picks up the secondary stain, which is safranin, or the counter stain, and stains red. Saffron, it's a flower that's orange. If you had used saffron to prepare rice, the rice stains orange, kind of red-orange. So safranin, it's red in color, so it stains the gram-negative bacteria red. So here are the different steps of gram stain. So you use crystal violet, primary stain, and if you have a culture of bacteria or a specimen where you have gram-positive and gram-negative, both of them, they will stain purple, the gram-positive and the gram-negative in the first step. You put this dye, crystal violet, for 1 minute. Then you rinse, and then you add the mordant, which is going to be iodine. And then both of them will remain purple, the gram-positive and the gram-negative. The mordant you add it for 1 minute, iodine you add it for 1 minute. Then you rinse, and then you add a decolorizing agent, which is alcohol, acetone alcohol, for 10 seconds, and then you rinse. After that, gram-positive bacteria will remain purple, while gram-negative will remain colorless. And then you will add, in the last step, after you rinse the decolorizing agent, which is alcohol acetone, you will add safranin. And you leave it there for 1 minute. And then you rinse. And then, if you had gram-negative in your specimen, the gram-negative bacteria is going to look red, while the gram-positive remains purple. Why it remains purple? Because there is no space in the cytoplasm to add this. And you retain the crystal violet with the mordant, with iodine. You create a gigantic complex, which is crystal violet iodine complex. This technique, if it's well done, it will not take more than 5 minutes. And you use like 1 or 2 minutes more to observe it under the microscope. And then you can tell if that specimen, let's say if it was cerebrospinal fluid, then you can tell if the person had a bacterial infection with gram-positive or gram-negative. Who cares? Everyone should care about it. It can be a kid with meningitis, it can be a college student with meningitis, and a 5-minute technique can save a life, because meningitis is an infection of the meninges in the brain, and it can leave the person unconscious, in coma, or with neurological damage. Because with the appropriate technique and diagnosis, you can give the appropriate treatment. So this technique is very important. And here's the steps again. So you add crystal violet, keep it there for 1 minute, rinse with water, add the mordant, the iodine, keep it there for 1 minute, rinse with water, add the decolorizing agent, 10 seconds, rinse with water, add the counter stain, leave it for 1 minute, rinse with water, let it dry, air dry, and then look at it in the microscope. Safranin is the secondary stain. So gram-positives and gram-negatives both will look purple up to this stage. And then only the gram-positive will stay purple all the way, while gram-negatives, if they exist in the sample, will be colorless after decolorization and will remain colorless unless you add safranin. If you don't add safranin, you have missed the opportunity of find out the gram-negatives. Let's say it's a technician is working on the weekend, Sunday night, Saturday night, in basically 3 a.m. It runs out of safranin. It receives a sample of CSF to stain it. It runs out of safranin. And it sees only gram-positive bacteria. Can this technician in the lab just write down, well, the CSF is contaminated with Staphylococcus aureus, which is a gram-positive bacteria? Can it say that unequivocally, I only found this? If it was a CSF of one of your cousins, little cousins, or maybe your son, your daughter, it's a baby, would you like that, to not add safranin? I don't think so. That's why even this last step is very important, guys, because you can detect something that might be missing if you don't add the safranin, and you can save a life. And this is how it looks, a mixed culture in the microscope. So you have gram-negative rods and gram-positive cocci. Rods and cocci are shapes of bacteria. We will see later on that. But basically, gram-negatives sustain red, gram-positives purple. So that's the most common technique that we use for staining. But we also have another one that is called acid-fast. And acid-fast, it has some variations like Ziehl-Neelsen. And in this case, we use carbofuchsin, which is basically a red dye that stains acid-fast microorganisms red. And the non-acid-fast will stain with the counter stain, which is methylene blue, which is the counter stain, and they stain blue. So this technique uses a decolorizing agent, which is acid alcohol. So it's alcohol with hydrochloric acid. And then the microorganisms that you try to stain with this technique are acid-fast microorganisms like nocardia, like Mycobacterium. And then you put your sample, your smear, on top of boiling water. You put filter paper on top of this, and you will be adding first carbolfuchsin for 5 minutes as this water is vaporizing under the glass slide. That is going to warm up your specimen, and it will open the wall of the bacteria. So because this bacteria has very thick layer, typically, like in the case of Mycobacterium, of a substance that's called mycolic acids. It's like a waxy material. The dyes, they don't penetrate as easily, so you don't use gram stain. So you use then this carbolfuchsin, and you float this carbolfuchsin on the top of this filter paper, and with the heat, with the warm vapor as it's going through, it will make these walls to open up a little bit. Carbolfuchsin goes inside, and then if this bacteria is actually acid-fast, in the decolorizing step, it will retain this carbolfuchsin. If it doesn't, it will lose it, and then that's why you add the counter stain, which is methylene blue, which will make it keep it if it's a non-acid fast. And in this case, you keep this sample in vaporizing water with carbolfuchsin for 5 minutes. Then you withdraw it from there. You take off the filter paper, you rinse the excess of carbolfuchsin, now you add the decolorizing agent, and then you rinse it, and then you add the counter stain, you leave it for 1 minute, and then you rinse it again, and then you let it air dry, and you look under the microscope. So acid-fast will stain red, while non-acid-fast will stain blue, and these are the steps. If you have non-acid fast, like in the case of the right side column, and acid fast in the left side column, both of them in the first step will stain red. Then second step, which is decolorizing, only acid fast will stain red, will remain red, while the non-acid fast will be colorless. And then the last step, only the non-acid fast will stain with blue because it doesn't have this carbolfuchsin at cytoplasm. And this is how it looks under the microscope. So Mycobacterium bovis, this bacteria is acid-fast, so it will stain red, while these other spheres, or this cocci, is non-acid-fast, so this will stain blue. Any questions? So we will leave it up to here, and we will continue next.